Well, I come from math. Thank you. Appreciate it. All my friends from Shack, that's why you see that applause, but really appreciate it. I come from a math and quantum physics background, and I have 25 years in AI research. I did, MIT OCW online in terms of cellular and molecular biology only during COVID because I was like, why is a virus shutting down world economies for like 2 years, right? Either way, that's a segue into me trying to think about the problem of life sciences or clinical biology from first principles. And I've heard things about, we need more data, we need more hardware. But I'm going to give you a contrarian view, and I'm also going to tell you what we're doing at Cognet, right? So the first assumption was 95% of your trials fail in phase 1 and phase 2, primarily because of on-target and off-target toxicity or efficacy. And your question is, but why does it do so? And you go back to, well, in the preclinical phase, you don't have human models which actually replicate what happens in vivo. Fair enough, but there's a lot of in vitro, cells that's actually available during preclinical. Then when you start looking at that assumption, the efficacy of these cells, because of how you go ahead and immortalize them, actually loses its stochastic variant on what really happens in vivo. So I said, okay, this is one constraint that you have to work with. Second, when people talk about data, the basic question when you think about AI is that how large should the data be? And most importantly, what should be the variance in this data that statistically captures enough detail and not more or not less that actually allows you to actually learn something about the behavior, right? So these were some of the first principles that I was thinking about. And I was also thinking about, well, if you want to really decipher the language of life, can we take the approach of human language? Like you go ahead and stick your K-mers into a transformer, something should come out, you should have a novel biological sequence. So worked on it for 8 months and it's absolutely horrible for many different reasons. And the challenges are this, that you're actually working with 3.2 billion base pairs, each having 4 characters. That's 4 raised to the power of 3.2 billion possible variabilities. And on the druggable candidates, you're working with 10 to the power of 60 possible druggable candidates. Added to that, you have epigenomes where there's 100 different post-translation modifications just for your histone on the long tail in terms of acetylation and methylation. So this adds complexity in terms of the infinite search space for you to actually even find a dose-dependent pharmacodynamic biomarker which are druggable. But what kind of drugs? Are you going to apply gene therapy? Are you going to apply CAR-T? Are you going to apply biologics? Is it going to be, immunology? So that becomes a challenge, right? So I went back, in physics, you don't ask about how much data is available. You think about first principles in terms of what's the governing function, which actually encapsulates the behavior for me to approximate a solution. So we said, okay, here's a great opportunity for me to actually work on a horizontal platform, which is what Cognet is all about. What we do is we take, we learn across joint distribution across eukaryotes, which has enough variability for you to learn something about every gene, its biological pathways, and its histones and chromatin modelers, protein, DNA interaction, and so on. So if there's enough variance, I'm going to learn cross-cell, I'm going to learn cross-species to understand how this multi-grammar, orchestration about what we call as the language of life works with each other, and does the foundational model that you have, either a diffusion model or like a large language model, allow you to actually capture this. So the first answer was easy. Geo, ENCODE, Phantom over the last 15 years have made available data that is publicly available. They're noisy, you have to do a lot of imputation, so we can work with it because that's pretty much a math and a physics problem. That's not really a biology problem or a data or a hardware problem. So start looking at that, and then we said, okay, the second challenge is we don't have any model which actually enables multi-grammar. It's not multi-model, the language is made of multi-grammar. Your large language model is unigrammar model. You read from left to right and you understand everything. So that's how we created Cognet, where now we have an 83% accuracy in terms of simulate a gene regulatory network by just inputting a whole genome sequence, which outputs a biogrid, where on the x-axis you have 60,000 gene mutations, on the y-axis you can actually simulate 5 to 10,000 cell conditions. The cell condition can be, this is a female in a menstrual cycle, and this, the cell phase is so on and so forth, and you can apply treatment regimes, which gives you about 83% accuracy of what might happen in vitro. So what's the use case of this? The use case is you're not looking for faster horses, where you say you look for point solution by applying robotics or by doing robotic process automation. Instead, you say, given this large search space, can I quickly go ahead and find where the needle in the haystack is? I mean, everywhere else versus, can I say with 83% accuracy, if you do your experimental validation in this space, your probability of success increases. So that we have able to showcase. And one of my favorite protein, I think, is TP53. The P53 is called the guardian genome. And the use case that we have shown is how do you upregulate or downregulate. If you upregulate P53, which is a tumor suppressor gene, your neuronal activities is going to come down. That's the cause for your Alzheimer's and Parkinson's, right? So if you downregulate it, that means you're actually causing this Alzheimer's and neuron. But if you upregulate it, you'll actually have problems in terms of oncogenes taking over and creating tumors, right? So these are some of the use cases that we have showcased. And with this new wave of technology, you're not looking for larger data, you're not looking for faster hardware. You're now able to have a conversation with your genome to say, express yourself. I want to talk to you. Tell me who are your friends are, who are your enhancers or promoters. And this allows us to get to the new future where you don't have to spend 12 years, $2.9 billion. You don't have to sacrifice 250 million animals year on year in the name of science by moving a large amount of this protocol to in silico. Thank you.